So, good morning, good afternoon, or good evening, according to your time zone. I welcome you to this seventh lecture of the online training series, Observing Marine Ecosystem Structure and Function, from Microbial Communities to Fisheries and Human Health. This is the virtual component of the in-person training course going to be held in India at the end of October, preceding the TREVOR Symposium. Apart from the students going to attend the in-person course, this series is open to the whole marine research community, especially encouraged our students and early career professionals. So we are happy to have a large audience today. Again, a reminder that this is a lecture, so take advantage of the knowledge of the speaker and the panelists, and post as many questions as you want, and please post them on the Q&A box, and we'll have plenty of time to get to them after the talk. Before introducing our speaker, we have also a panel to contribute to the discussion, and it's composed by Dr. Shubha Satyendranath, a senior scientist at the Plymouth Marine Laboratory in the UK, also Dr. Chima Koik, also senior scientist at PML, and Dr. Priscilla Lange, research and innovation manager at the National Institute for Ocean Research in Brazil. Hi, Priscilla. And now it's my pleasure to introduce our speaker, Dr. Ryan Vandermeulen. Ryan currently serves as the Satellite Program Coordinator for the Office of Science and Technology at NOAA Fisheries, where he oversees the integration of satellite data into fisheries management and ecosystem monitoring. A self-professed ocean color enthusiast, Ryan has developed algorithms, contributed to the planning and validation of several ocean color satellite missions, and otherwise spent a considerable amount of his professional career pondering the meaning of life through the lens of phytoplankton. He got his CLEX during graduate studies at the University of Southern Mississippi, followed by collaborations with government research laboratories and extensive field campaigns along the US Gulf Coast. After 7 years supporting ocean color research and satellite mission activities at NASA, Ryan joined NOAA Fisheries, where he enjoys seeing how satellite data are applied in practice and helping translate complex observations into actionable insights for sustainable fisheries and ocean conservation. Ryan has an intriguing title for our talk today, Zen and the Art of Measuring Primary Production at Sea. Ryan, the floor is yours.
Thank you very much. Excellent. All right. Well, good afternoon or morning or perhaps late witching hour, wherever and whenever you are. Really appreciate you dialing in today. It is it really is truly an honor to be a guest in this lecture series, and I'm very eager to chat with you all today about how we quantify one of Earth's most fundamental life-enabling processes. I will say, when I was invited to speak on this topic, I thought, this is perfect. This is a topic I've spent a lot of time thinking about, and here is a chance to really consolidate my thoughts on the matter and put together a clean, comprehensive narrative about primary productivity measurements in the ocean. And then I went to sit down and build the talk and immediately realized what a foolish and problematic assumption that was, because oceanic primary productivity is one of those subjects where every answer seems to come with an asterisk, and frankly, even agreeing on exactly what we mean by primary productivity is not always as straightforward as it sounds. So at its simplest, primary productivity is the process by which photosynthetic organisms use energy from sunlight to convert inorganic carbon into organic matter. But when we're actually trying to measure this activity in the ocean, that simple definition can quickly branch into multiple possibilities. Are we talking about how much sun energy is captured by phytoplankton in the ocean, oxygen produced from photosynthesis, the amount of carbon being fixed into biomass, perhaps the amount of consumable energy available to the food web, or the total flux of atmospheric carbon dioxide that ends up in the deep ocean? The answer is yes to all of those. Those are all legitimate ways of thinking about primary productivity, and they're all describing different moments of the same story. So rather than structuring the lecture around some of the individual methods we use to estimate productivity, I thought it best to organize it around the process itself. So we're going to have a little fun today and follow the journey of a singular photon, starting from its very formation and through its interstellar journey across space, all the way to its arrival at Earth, where it enters the ocean and begins its interaction with phytoplankton. From there, we're going to trace that energy all the way through the photosynthetic machinery into the broader marine ecosystem and ultimately into the global carbon cycle. So along the way, we will stop whenever scientists have found a clever way to capture a piece of that story with some sort of measurement. And my hope is that by understanding the process first, the strengths and limitations and assumptions behind each of these various methodologies becomes more intuitive. All right. So at the beginning of our saga, we must acknowledge the illustrious heroine of our solar system, the sun. So the energy driving life in the ocean begins its story deep inside our brilliant star, 150 million kilometers away from Earth, long before it ever encounters a single molecule of chlorophyll. So every second, our sun converts about 600 million tons of hydrogen into helium. And in doing so, it releases an unfathomable amount of energy that a minuscule fraction of this is all it takes to power nearly all of life on Earth. So please indulge me as we take a moment to just appreciate this incredible journey. So deep in the core of the sun, gravity has spent billions of years compressing hydrogen into an environment that's essentially a giant natural fusion reactor. So at temperatures of around 15 million degrees Celsius, hydrogen nuclei are moving fast enough that occasionally they overcome their natural electrical repulsion and collide. So through a series of reactions called the proton-proton chain, hydrogen atoms are gradually fused together to form helium. And the mass of the final helium nucleus is slightly less than the mass of the original hydrogen nuclei. And that missing mass is then converted to energy. This is Einstein's famous E equals mc squared in action. Because the c portion, the speed of light squared, is such an enormous number, even a tiny amount of converted mass represents a tremendous amount of energy. So the energy begins as extremely high energy gamma radiation, and this is where our traveler is born. The energy packet that will eventually become the sunlight as we know it is about to undergo a very long and semi-chaotic journey. So our little gamma ray photon has officially entered the universe, and it can technically move at the speed of light, but it immediately gets stuck. The problem is inside of the sun is incredibly dense. There are so many electrons and atomic nuclei packed together that this energy can only travel about a centimeter before interacting with something. It gets absorbed and then re-emitted. It changes direction. It takes another step and does this again and again and again. And this is called the random walk. Some estimates vary, but this journey can take tens of thousands to hundreds of thousands of years before the energy finally reaches the surface. But after countless absorptions and re-emissions, that original gamma ray energy ends up being shared among many new lower energy photons. Now, eventually, this redistributed energy reaches the surface of the sun, or the photosphere, and here the density drops dramatically, the path clears, and a broad spectrum of sunlight, dominated by visible and infrared light, finally escapes into space. So from this point on, we're going to follow one representative visible light photon carrying a small portion of that energy. After, we've now gone over hundreds of thousands of years being trapped inside the sun, and now this photon is barreling through the vacuum of space at exactly 299,792,458 meters per second. And 8 minutes and 20 seconds later, it arrives at our blue planet. So as our photon approaches Earth, it encounters its next challenge, which is the atmosphere. You'll notice I have taken a few cutesy liberties with the animations here. Our photon is not actually a glowing comet streaking through the sky, but I think this is a helpful way to at least convey that sunlight arriving at Earth is not guaranteed to make it to the surface. The atmosphere filters incoming radiation. Some wavelengths are absorbed by gases like ozone and water vapor. Some are scattered, some are reflected back into space, and some photons are lucky enough to make it through the atmosphere. And today, our photon has a very specific destination. So finally, after a long and arduous journey, our photon has reached the ocean surface. Again, some creative liberties with the splashing effect, but I think our characters earn the right to be a little extra at this point. So at the air-sea interface, the photon faces yet another set of possibilities because not all sunlight behaves the same once it enters the ocean. Remember, sunlight is not just one single thing. It's a massive collection of photons, each carrying a specific wavelength and energy. And just like the atmosphere, the ocean also acts as a selective filter. So some photons are absorbed almost immediately by molecules in the surface ocean. Longer wavelengths like red light are absorbed very quickly by water molecules, while shorter wavelengths, especially blue light, are able to travel much deeper into the water column. And some photons are scattered by particles or molecules and change direction. Some are simply reflected back into space, and then some continue downward. And this is the conclusion of our astrophysics narrative and where our oceanographic journey officially begins. Now, this here is a special moment, ladies and gentlemen. Our photon has spent its entire existence trying to get somewhere useful, to find a sense of meaning and purpose. And here it has stumbled upon a tiny photosynthetic diatom drifting somewhere in the Pacific Ocean. The connection here is near instantaneous, and with a crooked smile from our photon friend, we are invited into this silica-based housing surrounding the cell. It's once our photon enters the cell that it encounters the photosynthetic machinery. So the key player here is chlorophyll A, the molecule that captures light energy and starts the process of photosynthesis. Now, chlorophyll A is not the only pigment in town. Phytoplankton carry a whole suite of accessory pigments, including carotenoids or pigments like fucoxanthin that help expand the range of wavelengths they can capture. So our diatom is a pretty sophisticated light harvesting system. But when a photon is finally absorbed, its energy is then transferred to electrons within the pigment molecule, moving them into an excited state. Now, the best case scenario is that the excitation energy is transferred into photosynthetic reaction centers where it drives the electron transfer reactions that ultimately convert light energy into chemical energy. We'll talk about that more. But not every photon gets that opportunity. Some absorbed energy is immediately dissipated as heat. This is actually an important protective mechanism because too much absorbed light can damage photosynthetic machinery. And a very small fraction takes a third path, fluorescence. So fluorescence occurs when an excited chlorophyll molecule returns to its original lower energy state by releasing some of that energy as a new photon. So an emitted photon has a longer wavelength than the original one because some of that absorbed energy has already been lost through other processes. This physically manifests as a tiny red glow, and while it represents only a small fraction of the absorbed energy, it is something we can measure. And this brings us to our first measurement approach, the classic in situ fluorometer. So for the remainder of this talk, every time we introduce a new method or instrument, I'm going to use sort of a common framework here. What does it measure? How does it work? What are the advantages and limitations? And if you look along the bottom, we'll keep track of where we are in the journey because every method is observing a different part of the same process. Fluorescence gives us our earliest glimpse into this pathway. It tells us that light has been captured, and with some context, it provides information about the state of the photosynthetic machinery. However, it doesn't directly tell us how much carbon is being fixed just yet. So what a fluorometer does is it shines blue light into the water, which excites any proximal chlorophyll molecules, and then an adjacent detector measures the emitted fluorescence. Generally speaking, more chlorophyll in the cell equates to more potential fluorescence, and with careful calibration, we can use the signal to estimate chlorophyll concentration. The major advantage here is speed. These instruments are small, inexpensive, and can collect measurements continuously from ships, autonomous platforms, and moorings. The limitation is that fluorescence is an indirect proxy measurement. The signal not only depends on how much phytoplankton are present, but also their physiological state and their recent light history. For example, under high light conditions, phytoplankton can activate a protective mechanism called non-photochemical quenching, or NPQ, where excess absorbed energy is safely dissipated as heat rather than being passed through the photosynthetic pathway. So this changes the relationship between fluorescence and photosynthetic activity, meaning that the same exact amount of chlorophyll can actually produce different fluorescence signals depending on the conditions of the cells. But if we want to know how much of that captured energy has the potential to be converted into new organic carbon, we need to follow our photon a little further downstream. So the goal of photosynthesis is not simply to capture light. The goal is to convert that light energy into chemical energy. And that process begins at photosystem 2. So photosystem 2 is one of the first major protein complexes in the photosynthetic electron transport chain. So when chlorophyll absorbs a photon, that energy excites an electron to a higher energy state. And those electrons then enter an electron transport chain where the movement ultimately helps create the chemical energy needed to build organic carbon. But those electrons have to come from somewhere. So photosystem 2 solves this problem by splitting water molecules. So here, two molecules of water are broken apart, releasing electrons to place those lost from chlorophyll and then producing oxygen as a byproduct, as well as contributing protons that help generate the energy gradient used by the cell. So now our photon has completed its first major task. It has been converted into electron flow. And believe it or not, there's a way we could actually watch an aspect of this process happening. So this brings us to active variable fluorescence. Now we're in the territory of examining how efficiently the photosynthetic machinery is using the light energy it receives. So the instrument works by sending really rapid pulses of light at a water sample. So these pulses temporarily close the reaction centers of photosystem 2 by basically filling the available electron acceptors. So when photosystem 2 is closed, fluorescence increases because that absorbed energy can no longer be efficiently directed into photochemistry. As we measure this, the difference between the minimum sort of background fluorescence and the maximum fluorescence, FM, gives us the variable fluorescence, or FV. When normalized to the maximum fluorescence, this provides a window into basically how efficiently photosystem 2 is converting absorbed light energy into electron flow. In general, a higher sort of FV to FM ratio indicates healthier, more efficient photosynthetic machinery, while lower values tend to suggest that photosystem 2 might be stressed or that absorbed energy is being diverted away from photochemistry. So after the light pulse, the fluorescence signal relaxes as photosystem 2 returns to its functional state. The speed of that recovery also provides additional information about how the organism is processing and dissipating absorbed light energy. Because electron transport supplies the energy and reducing power required for downstream carbon fixation, these measurements can be combined with physiological models to estimate gross primary productivity. So this is our first type of primary productivity. Gross primary productivity represents the total amount of carbon fixed by photosynthesis before accounting for any energetic losses further down the pathway. So the advantage to this methodology is that these measurements are fast, they're not destroying the sample in any way, and they can be collected continuously for really high resolution insights. There are, however, some pretty hefty assumptions that have to be made about how electron flow is actually allocated towards carbon fixation to get at that assumption about carbon fixation. All right, so far our photon has excited the chlorophyll molecules, driven an electron transport chain, and ultimately reached one of the defining reactions of photosynthesis, the splitting of water. And this gives another opportunity. Instead of following the photon energy itself, we can follow one of the products it leaves behind, oxygen. Recall here that the splitting of water molecules provides the electrons needed to replace those lost from photosystem 2, and then oxygen is released here as a byproduct. So one way we can quantify this process is to collect a seawater sample, enrich it with oxygen 18 labeled water, and then incubate it under simulated light conditions. So oxygen 18 is a naturally occurring heavier form of oxygen that we could use as a tracer. Because the oxygen released during photosynthesis comes from water, any oxygen produced from the oxygen 18 labeled water carries that same 18O signature. So by measuring the isotopic composition of the dissolved oxygen pool, we can directly quantify how much oxygen was created through this specific photosynthetic process. We call this gross oxygen production, or the rate at which photosynthesis is generating oxygen through light reactions. This is an important distinction because gross oxygen production is not a measurement of carbon fixation itself. It's a measure of a different currency, oxygen instead of carbon. But because oxygen production occurs in the same light-driven machinery that supplies the energy and reducing power for carbon fixation, they're very closely linked. With the right assumptions about the relationship between oxygen evolution and carbon fixation, this provides a really powerful way to estimate photosynthetic production. The major advantage here is that we are measuring a direct product of photosynthesis. The trade-off, though, is this requires specialized isotope measurements, typically using mass spectrometry. We also have to consider whether the bottle we've put our sample in accurately represents the natural ocean. The moment we put take seawater out of the ocean and put it into a bottle, we have changed a variable. We have changed the environment. We've removed it from currents, altered light conditions, and potentially changed the community that we're trying to measure. So what if we can get the same information without the bottle? And this is where triple oxygen isotopes come in. Instead of adding a tracer, we can use the natural isotopic fingerprint that's already present in dissolved oxygen. So oxygen exists naturally in three stable isotope forms, 16O, 17O, and 18 oxygen. So photosynthetic oxygen production leaves behind a very distinctive isotopic signature because oxygen, again, originates from water molecules. And by measuring these isotope ratios in seawater, we can actually separate oxygen produced by photosynthesis from the pool of oxygen that was exchanged with the atmosphere or modified by respiration. The major advantage is that this provides an estimate of gross oxygen production directly in the environment without requiring incubation bottles. Samples are relatively simple to collect, allowing measurements across large spatial scales. And because this method kind of integrates the natural environment, it avoids some of the assumptions associated with laboratory incubations. The challenge is that the subtlest signal, though, requires an extremely precise isotope, measurements, and physical processes like mixing and transport can complicate the interpretation a bit. And again, oxygen production must, ultimately be related back to carbon fixation if we want to talk about a carbon budget. And the ratio of oxygen produced to carbon fixed is known as the photosynthetic quotient, and it's not necessarily a one-to-one ratio, and it's not strictly constant either. There are some uncertainties to consider there. So at this point, we've followed the photon through several stages. We've measured the light harvesting machinery with fluorescence. We've measured electron transport. And now we've measured the oxygen released when water was split. But eventually we come back to the question, how much carbon did phytoplankton actually create? And for that, we need to follow the carbon itself. So let's return to our photosynthetic machinery here. Up until this point, we've been following light reactions, the part of photosynthesis where sunlight is captured and converted into chemical energy. But photosynthesis is really two connected processes. The first stage captures energy, and the second stage uses that energy to build carbon. So here we are looking at the transition between those two stages. When our photon was absorbed by chlorophyll in photosystem 2, it started a cascade of electron transfers through the photosynthetic electron transport chain. As those electrons move through the chain, they help create a proton gradient against the membrane. It's kind of like the concept of a hydroelectric dam. The cell uses that stored energy to make ATP, essentially the energy currency of the cell. At the same time, electrons eventually reach photosystem 1, where they are used to generate another important molecule, NADPH. So ATP and NADPH are now carrying the energy and reducing power that the cell needs for the next step. And this is where our photon story comes to an end. The photon itself is gone, its energy has been transformed into chemical currency, and the cell has effectively converted sunlight into a form that it can use to build something. So that something is ultimately going to be carbon. Now, if you're starting to get the sweats and are having flashbacks of your undergrad graduate biology courses, please try to remain calm. I promise I will keep this high level. But the Calvin cycle is where the cell takes inorganic carbon dioxide from the environment and transforms it into organic carbon. And the key first step is carbon fixation, taking carbon that is floating around as CO2 and incorporating it into a molecule that the cell can use. So the enzyme responsible for this first step is Rubisco, arguably one of the most abundant proteins on Earth. It is also infamously known as one of the least efficient enzymes on Earth. This is a bit of a distracting sidebar, but it is good trivia knowledge. So many enzyme processes, many enzymes will process thousands of substrate molecules per second. Rubisco is often only fixing a few CO2 molecules per second. It can also occasionally accidentally grab oxygen instead of carbon dioxide, triggering a process known as photorespiration, essentially undoing some of the work that the cell just did. The reason for this is that Rubisco evolved in an ancient atmosphere where CO2 was more abundant and oxygen was lower. So modern organisms basically just inherited this molecular compromise. But when you consider how much carbon the ocean and terrestrial ecosystems fix every year, it gets the job done. Okay, sidebar done. So using the ATP and NADPH generated from the light reactions, the Calvin cycle converts that fixed carbon into organic molecules. We now have reached carbon fixation. This is where one of the most historically important measurements in biological oceanography enters the story. So in 1952, Danish oceanographer E. Steeman Nielsen introduced a method that fundamentally changed how we measure primary productivity. His idea was elegantly simple. What if we could just label the carbon dioxide available to phytoplankton, allow photosynthesis to proceed, and then measure how much of that labeled carbon ended up inside organic matter? So to do this, seawater samples were collected into glass bottles, enriched with radioactive bicarbonate or C14, and then incubated under controlled light conditions. So after the incubation, the amount of radioactive carbon incorporated into particulate material can be measured. Now, phytoplankton are not all living under the same light conditions. So a cell living near the surface experiences intense sunlight, while a cell deeper in the water column experiences much less light. So rather than just one bottle, we can create a series of controlled light environments. So this is the idea behind the photosynthetron, a device here that allows us to expose samples to a gradient of different light intensities and then measure how carbon fixation responds accordingly. So combining this photosynthesis light relationship with information about the underwater light field and the daily cycle of available sunlight, we can then use this to estimate how much carbon is fixed over the course of an entire day throughout the water column. Now, there is an important distinction to make here. Photosynthesis creates organic carbon, but cells are constantly using some of that carbon for their own metabolism through respiration, which we'll talk about next. Now, gross primary production is everything fixed by photosynthesis. By contrast, net primary production is what remains after subtracting phytoplankton respiration. So this carbon-14 method is interesting because where it falls on that spectrum depends partly on how long the incubation lasts. During short incubations, most of the newly fixed carbon remains associated with the photosynthetic machinery, so measurements tend to be closer to gross production. Over longer incubations, over a 24-hour period, some of that carbon is redistributed, it's respired, or it's released as dissolved organic carbon. So the measurements increasingly reflect something closer to net production. While there is some uncertainty here, the important thing is that we're finally measuring carbon. This is an extremely sensitive method. It works even at very low productivity rates in oligotrophic gyres and has generated one of the longest and most valuable productivity records in oceanography. But as we mentioned, the interpretation can be somewhat fuzzy, and we still have to contend with the fact that we're physically moving phytoplankton out of their native environment. Okay, so we've got a steady stream of carbon income coming into the cell thanks to our friendly photon. But like every living organism, phytoplankton have operating expenses. They have to maintain their cellular machinery, repair damage, acquire nutrients, maintain ion gradients, all this just to stay alive. So to pay that bill, as it were, phytoplankton respire. So some of the organic carbon they just work so hard to build is broken back down. Oxygen is consumed in this process, ATP is generated, and some of that precious carbon ultimately leaves the cell again as carbon dioxide. So to kind of break it down, where photosynthesis generates income, respiration represents our basic expenses. So a lot of that carbon is not burned and is essentially invested into growth. Some of that carbon becomes proteins, some of it lipids, DNA, some of the carbon builds new membranes and cellular machinery. Eventually, all of those pieces come together to make new biomass, new phytoplankton cells. And we call this net primary productivity. It's the portion of carbon fixation that remains available to support growth after the cell has paid its metabolic cost. So ecologically, it's a pretty significant part of the primary productivity continuum because it is essentially a step where new food becomes available to the marine food web. Just a quick recap, so we're covering a lot of terms. So far, we've measured gross primary productivity in a few different ways, which represents our total carbon income. And now we will measure net primary productivity again, so this is what's left after paying bills. So radioactive carbon-14 remains an incredibly sensitive tracer and a great way to measure net primary productivity. But working with radioactive materials comes with some logistical challenges. Ships need dedicated radioisotope labs, specialized handling procedures, and strict waste protocols. Some ships just strictly won't let you on board with C14. An increasingly common alternative is to replace radioactive carbon with its stable isotope, carbon-13. The experiment itself is nearly identical. We add 13C labeled sodium bicarbonate to seawater samples, and then we incubate them under carefully controlled light and temperature conditions, and then measure how much of that labeled inorganic carbon ends up in particulate organic matter. Now, on deck, we can recreate the light environment using flowing seawater for temperature regulation, as well as neutral density screens to simulate different depths. The major advantage here is control. We can carefully regulate temperature and light and repeat experiments very consistently and importantly, avoid having to handle radioactive materials. The trade-off, though, is sensitivity. Since we are measuring a subtle shift in the natural abundance of carbon isotopes rather than radioactive decay, larger sample volumes are typically required, and the analysis requires specialized mass spectrometers, which tends to be a little more pricey in terms of the equipment that you need. Another alternative that works with either C14 or C13 labeling, we could simply return the bottles back out to sea after collecting water at the same depth that they were collected, and then incubate them under the natural light field and ambient temperature experienced by phytoplankton. In many ways, this is the closest we can come to letting the organisms kind of continue their normal day without making a controlled, while we're still making a controlled measurement. By allowing samples to experience the natural underwater light field, they're experiencing changes in cloud cover and solar angles, spectral quality in a more realistic sense. So we reduce some of the artificial conditions that inevitably accompany on-deck incubations. Of course, the bottles still isolate the cells from their surrounding ocean, so it's not 100% natural. There's still these kind of bottle effects to contend with, and respiration and dissolved organic carbon are still being released and kind of complicate the interpretation. And perhaps one of the more important logistical considerations, because the bottles remain in the water for hours, each station requires a substantial investment of ship time. So whether we're in the lab or on deck or back in the ocean, regardless of which method we choose, there's all legitimate ways of measuring carbon uptake, but the difference is really how much realism, control, sensitivity, and logistical complexity we're willing to trade. Now, I'm not going to focus too much on satellite methods today, because our focus today is on field measurements to underpin our understanding of primary productivity, but it's worth briefly pointing out where satellites fit into this story. Now, in general, satellites are trying to estimate this specific part of our journey, net primary productivity. Of course, satellites don't measure primary productivity itself. They measure the color of the ocean, from which we estimate properties like chlorophyll concentration or phytoplankton carbon or phytoplankton absorption. We then combine those observations with information about available sunlight, light penetration through the water column, and temperature and mixed layer structure into photosynthesis models that estimate the production throughout the sunlit portion of the surface ocean. The obvious advantage here is scale. This allows us to study seasonal scales, interannual variability, long-term trends, and ecosystem phenology in a way that's really not possible to do from ships alone. The trade-off is, though, that these are model-based estimates. Their accuracy depends on both the quality of the satellite retrievals and the assumptions built into the productivity model, particularly how phytoplankton physiology and the vertical structure of the water column are represented. So while satellites give us extraordinary coverage, they ultimately remain connected to the field measurements we've been discussing. Those measurements are what allow us to build, improve, and evaluate these models in the first place. All right, moving on, our journey is far from over. Our dear photon has served us well, but from here on out, we're going to continue our journey through the lens of our new carbon that we've created. Equally as cute as the photon, I'll say. I've given it rosy cheeks, but now, we've been talking about a different form of currency, carbon. But a cell is not able to build itself from carbon alone. Those carbon skeletons need to be combined with other essential elements, particularly nitrogen and phosphorus. They need this to build proteins, nucleic acid, pigments, and all the other components of a living cell. So this process of incorporating nutrients into organic molecules is nutrient assimilation, and it is what ultimately transforms fixed carbon into new biomass. Now, unlike sunlight, these nutrients are finite resources in the surface ocean here. Because of this, once these nutrients are incorporated into biomass, they begin, circulating through one of the most efficient recycling systems on Earth, the microbial loop. So within this loop, phytoplankton are grazed by tiny zooplankton. Organic matter leaks into the water as dissolved organic carbon. Bacteria consume this material, they decompose it, and they recycle nutrients back into forms that phytoplankton can use again. So one of the most important recycled nutrients produced through this process is ammonium. Because ammonium is already in a chemically reduced form, phytoplankton can assimilate it directly without first expending energy to reduce it. So this makes it a very efficient fuel for continued production. So this is what we call regenerated or recycled production. The ecosystem is largely running on nutrients that have already been used once before. Contrast this with a scenario in which new nutrients arrive from outside of that recycling loop. In the open ocean, this can happen through upwelling or vertical mixing, which brings deep, nutrient-rich water to the surface. These new, if you will, nutrients are supplied primarily in the form of nitrate and to a lesser extent nitrite. Unlike ammonium, nitrate must first be chemically reduced before phytoplankton can incorporate it into biomass. This requires an additional investment of energy. In some regions, specialized microorganisms perform this remarkable feat of taking atmospheric nitrogen gas and actually converting it into biologically available nitrogen, which is known as nitrogen fixation. Either pathway, suddenly the ecosystem has access to a fresh supply of nutrients. And if all the ingredients are available in the right proportions, this can set up for a major bloom of growth to occur. Because this production is supported by newly incorporated nutrients rather than recycled ones, we refer to this as new production. For fisheries oceanographers, this distinction is particularly important. New production represents a fresh input of nutrients that can support additional phytoplankton growth, increasing the potential for energy to move beyond the microbial loop into zooplankton, forage fish, and ultimately into species we may care about managing. And indeed, we can measure this too. So this approach is very similar to the carbon isotope methods we've been discussing, but instead of labeling inorganic carbon, we label the nitrogen source itself. We can add either 15 nitrogen labeled nitrate or 15N labeled ammonium to seawater samples and measure how much of that nitrogen is incorporated into particulate organic matter. The important distinction is where the nitrogen came from. Again, nitrate in the surface ocean is generally supplied from outside the immediate recycling of organic matter, through mixing or upwelling from deeper water. So nitrogen taken up as nitrate is used as an indicator of sort of a proxy of new production. Ammonium, on the other hand, as we mentioned, is commonly produced when the organic matter is broken down and nitrogen is being recycled in the ecosystem. So ammonium uptake is therefore used as an indicator of regenerated production. There are caveats, of course, associated with this. These are still bottle experiments. We're also introducing a tracer that can potentially influence uptake rates, and some recycling can occur during the incubation itself. Nevertheless, these measurements provide really valuable insight into the sources of nitrogen supporting phytoplankton growth, and by extension, the potential for carbon export and energy transfer through marine ecosystems. So the actual creation of carbon is only part of the primary productivity continuum. The ocean is not a storage warehouse where phytoplankton simply accumulate carbon forever. Carbon has places to go. It can be recycled by microbes, it can be exported to the deep ocean, or passed along through the food web to larger organisms. So now we're going to follow what happens after our phytoplankton have done the hard work of making new biomass. So for many organisms, the first major step is grazing. Tiny zooplankton consume phytoplankton, and that carbon is either incorporated into new zooplankton biomass or used to power their own metabolism. Just like phytoplankton, zooplankton also have metabolic costs. They need energy to swim, find food, reproduce, maintain cellular machinery, and just simply stay alive. So that energy comes from respiration. So we're consuming organic carbon as well as oxygen. Thus, every transfer through the food web comes with a metabolic cost. What this means is not everything that zooplankton consume gets passed along to the next level of the food web. This is often summarized as a roughly 10% rule. It's not universal law, and the ratio can vary quite considerably, but it provides a useful approximation that for every 1,000 units of energy entering as phytoplankton biomass, roughly 100 units become new zooplankton biomass. And while the majority is lost through metabolism, respiration, and other processes. To complicate these things further, the efficiency of this transfer is not fixed. And here's where the environment really matters. In colder waters, metabolic rates tend to be lower. So organisms snacking on phytoplankton might not require as much energy for maintenance, which allows for a greater fraction of consumed carbon to be allocated towards growth and reproduction. While in warmer waters, metabolism generally increases, so organisms can burn through energy faster, which can increase the amount of primary production consumed simply to maintain themselves rather than being converted into new biomass. This is an important ecological concept because this means that phytoplankton abundance is not solely controlled from the bottom up. Light and nutrients, definitely they determine how much production is possible, but consumers can also regulate phytoplankton from the top down. A productive ocean does not necessarily mean a lot of phytoplankton sitting around. Sometimes it means a very efficient food web that is rapidly consuming and recycling that production. So if we want to understand the fate of newly produced carbon, we can't just consider how much carbon the phytoplankton create. We also have to consider how much is surviving grazing, how much is respired, and how much remains available to move through the ecosystem. And one way to answer that question is directly observe the interaction between phytoplankton and their consumers. So with grazing experiments, we could take natural phytoplankton communities and compare treatments with and without grazers or with different grazer densities. And by tracking the changes in phytoplankton abundance or growth rates, we can estimate how much production is being consumed by zooplankton. This cool little contraption here on the right spins the bottles around really gently so the phytoplankton don't fall into tiny little pockets in the corners of the bottles, which would kind of set up an artificial feasting zone for grazers. But the advantage of this approach is that it directly connects primary production to the food web. We are physically quantifying how much of that created carbon is being transferred into another organism. Now, of course, you may have noticed this theme. The moment we put organisms into a bottle, we are simplifying the very interactions we are trying to study. And grazers can behave differently. Predator-prey interactions are constrained, and capturing the natural variability of the ocean always remains a challenge when we're talking about bottle experiments. Another way to look at this process is by tracking the energetic byproducts. Every organism in the system, phytoplankton, zooplankton, bacteria, and everything else, is consuming organic carbon and using it for metabolism. And that metabolic activity leaves behind a measurable fingerprint, oxygen consumption. For decades, one of the primary ways we have measured community metabolism is through changes in dissolved oxygen. And the idea is simple. In a light bottle, photosynthesis produces oxygen while respiration consumes it. In this case, these processes are happening simultaneously and balancing each other out to some degree. So we call this net community production. Now, if we suddenly cut off light to a bottle, put it into a dark bottle, photosynthesis ceases completely, but respiration continues. So the net decrease in oxygen tells us how much oxygen is being consumed by the entire community. So this is really the metabolic heartbeat of the ecosystem. Now we have two rate measurements: the total oxygen consumed in the dark, and then the net oxygen balance that occurs in the light. If we add those two rates together, we now have an estimate of gross oxygen production, which is roughly proportional to the total initial amount of carbon fixed from sunlight. So one method, 3 windows into the inner workings of the ecosystem: how much oxygen is being produced, how much is remaining, and how much energy the community is consuming. The challenge with traditional oxygen measurements, though, is that it gives us a few snapshots in time. Kind of have to take incremental samples, and we have to assume that the rates of production and consumption are constant over the course of an incubation. So we draw a straight line between point A and point B and assume a constant rate. But conditions are constantly changing, and so does the real-time response of the biological community. Using oxygen-based sensors known as optodes, we can actually watch oxygen production change continuously. To the right, I'm showing an experiment that monitored oxygen fluxes over the course of several hours in a very turbid estuarine environment. So this particular incubation device was left floating in the surface ocean, exposed to completely natural light conditions, up until the point shown by the vertical dashed line here, when the apparatus was covered with aluminum foil, and we get a measurement of total community oxygen consumption. The dark yellow line you see is showing the amount of light reaching the incubator, and the fluctuations you see are actually holes in the clouds and sunlight occasionally shining through to hit the surface of the ocean on this cloudy day. So the black line is tracking the corresponding oxygen flux. And you can see not only just the relative balance of production to consumption shift as the day goes on, you can kind of see a gradual change, but on shorter time scales, minute to minute time scales, those little fluxes of light creeping through the clouds elicit an instantaneous response from the phytoplankton community. This has also allowed us to see something really interesting. When the incubator is first covered, oxygen consumption doesn't immediately settle into a steady respiratory state. There's actually this initial steep drop in many cases in oxygen consumption, phenomenon known as post-illumination respiration. This suggests that some oxygen-consuming processes are still winding down after the lights go out. Processes like photorespiration and the Meller reaction can temporarily continue to consume oxygen because they're tied directly to the photosynthetic machinery. This is a little in the weeds, but I just wanted to point out that the ocean is not a machine running at a constant rate. It's a very dynamic biological system, constantly responding to changes in light, temperature, nutrients, and interactions among organisms. Okay, let's zoom out. Up to this point, we've been following the fate of carbon at the level of individual processes for the most part, right? A photon enters the cell, carbon is fixed, nutrients recycled, energy is transferred through grazing and respiration. But ecosystems are not built from individual reactions. They emerge from the integration of all these processes over time and space. So this is really how primary production becomes an ecosystem. So carbon fixed by phytoplankton supports zooplankton production, which supports larger consumers, eventually transferring energy through the food web to organisms at the top. Of course, directly observing these type of transfers at ecosystem scales is pretty challenging. Outside of very targeted approaches like analyzing the stomach contents or tracking individual organisms, we rarely get a complete picture of who consumed what at any sort of large scale. So instead, what we can do is use measurements of ecosystem level responses to infer how energy is moving through this system. Now, to do this effectively and at scales that are relevant to ecosystem level questions, we're going to step away from following individual rates and instead look for the fingerprints left behind by the entire community. While we're at it, let's stop with this whole taking things out of the ocean and putting them into bottles and just measure the water directly. We've already established that oxygen is significant and a very meaningful trace of biological activity. But when we try to measure it outside of a bottle, we're now subject to changes that may be incurred from the physical environment itself, such as oxygen being moved around through mixing and circulation, as well as the significant flux of oxygen resulting from atmospheric exchanges. This is where dissolved argon gas becomes really useful. Argon behaves really similarly to oxygen from a physical perspective. It dissolves, it responds to temperature, and exchanges with the atmosphere in a similar way, but it is biologically inert. So by comparing oxygen to argon, we can essentially isolate the biological oxygen signal and estimate the net community production directly in the ocean. Unlike bottle incubations, this approach doesn't isolate a small sample from its environment. Instead, we can measure this pretty continuously with a mass spectrometer set up properly, and we can capture a very spatially detailed picture of the integrated metabolic signal of the surrounding system. But the trade-off here is scale. This is a powerful ecosystem level measurement. And even though we can collect individual measurements at a very high frequency, the measurement itself quantifies a very large scale integrated process. So the ecological significance of net community production at this scale is that it represents the balance between production and consumption at the entire community level. So positive net community production indicates that organic carbon is accumulating beyond what the microbial community is consuming, creating potential pathways towards export, benthic food webs, and the transfer of energy to higher trophic levels. We gain a tremendous amount of ecological context, but we do lose some of the ability to attribute that signal to a single organism or process or a moment in time. Historically, a lot of these measurements have relied on ships, which of course are highly capable platforms that provide incredibly detailed observations, but only at specific locations at specific moments in time. To understand how ecosystems change across the entire, oceanic regions, we need measurements that can stay in the water longer, travel farther, and collect observations continuously. This is where autonomous platforms have really become transformative. So one approach is to use optical sensors to track changes in the amount and characteristics of particles in the water column. Because suspended particles scatter and absorb light, measurements such as backscatter and beam attenuation provide a really useful proxy for particle concentration. So with appropriate calibration, these optical signals can be related to changes in a particular organic carbon, allowing us to estimate changes in carbon biomass over time and then infer rates of carbon production and loss. A complementary approach is to use oxygen as a tracer of ecosystem metabolism. So while the optical approach follows changes in the carbon pool itself by proxy, oxygen measurements follow the biological processes that create and consume that carbon. So in this example, the upper panel here shows that oxygen shows the oxygen fluxes, providing a view of the kind of metabolic balance of the ecosystem and the light and dark, while the lower panel shows the corresponding changes in particulate carbon inferred from the optical measurements. These are two different perspectives on the same biological system, one tracking the carbon itself and one tracking the oxygen consequences of the processes moving that carbon. Like all methods, there are plenty of uncertainties to go around. In this case, we have to try to quantify and correct for some of those physical transport mechanisms. We are also reliant on sensors that may start to drift over time, but this does allow us to have a more sustained observations over ecologically relevant scales relative to what a bottle gives us. So throughout the talk, we've been following light energy and carbon as it moves through increasingly larger scales. And our final question revolves around the ultimate fate of this carbon. We've covered the pathway in which carbon remains in the surface ocean, where it continues to be recycled through the microbial loop and food web. But some fraction begins this long journey, the slow descent from the surface to the deep ocean. And this is the biological carbon pump, the process that connects biological activity at the ocean surface with the long-term storage of carbon in the deep ocean. And while the amount of carbon exported from the surface is relatively small compared with the total global photosynthetic production, this process really plays an outsized role in regulating atmospheric carbon dioxide over long time scales. So dead phytoplankton, aggregates of organic material known as marine snow, zooplankton fecal pellets, these all sink through the water column. They transport carbon away from the surface. So the rate of this sinking particle flux is known as export production. The challenge is that this journey is not straightforward. Along the way, microbes and other organisms, they break down this material, they convert organic carbon back into dissolved inorganic carbon that can eventually return to the surface. Only a fraction that survives this process really actually reaches the deep ocean, where it can be stored for decades, centuries, or even longer time scales. Now, over sufficiently long time scales, the balance of carbon accumulation represented by net community production that we talked about, this should approximately reflect the amount of carbon available for export. In other words, sustained positive net community production creates the biological potential for carbon export. So in this way, net community production and export production are similar, but they're distinct, and that export represents the fraction of that accumulated carbon that successfully leaves the surface ocean. So how do we measure something that's happening hundreds or thousands of meters below the surface? One of the oldest approaches is surprisingly simple. We just capture the particles as they sink. So sediment traps are essentially underwater collectors that intercept sinking material at a known depth. And by measuring the amount of and the composition of the material captured over time, we can estimate the flux of organic carbon leaving the surface ocean. And this gives us one of the most direct measurements of export production, the actual transfer of carbon from the surface ocean toward the deep ocean. Another advantage is that we don't just measure, how much of the carbon is sinking. We can collect that material and analyze it itself, its chemical composition, its pigments, the isotopes, and its biological contents. This can give us some additional context. This is also, currents can also influence what enters the trap. So sometimes living zooplankton can kind of stumble upon an absolute smorgasbord of stationary food. So this is restrictive to measuring particle fluxes and doesn't capture the dissolved organic aspect as well. So, all right, we've covered a lot of ground. Vivian, I see you popping up. How am I doing on time? Am I running over here? I'll try to.
You can continue. Carry on, please.
All right. I'll try to wrap up here, but we've covered a lot of ground here. Just to have a lot of fun, kind of keep things straight. I generated some reference cards for the 5 major processes we talked about today, and each process tells us something different about the ecosystem and answers a different scientific question. Each has inspired its own family of measurement techniques. So gross primary production is the total amount of carbon fixed through photosynthesis before anyone has paid the bills. You can think of this as the initial deposit, the full amount of energy entering the biological system before respiration and other losses begin to subtract from that pool. And if you want to understand the capacity of the ocean to capture sunlight and convert it into biological energy, GPP here is the fundamental starting point. And subtract the phytoplankton, its own expenses, the phytoplankton's expenses from GPP, and you're left with net primary production. And this is the new biomass that phytoplankton can invest in growth and reproduction, the material that can really begin to support food webs. And then community respiration is the ecosystem's operating cost. And every organism, phytoplankton, bacteria, zooplankton, uses energy to stay alive. And respiration is just telling us how much of that newly fixed carbon is immediately being utilized or spent. And then you subtract off the metabolic costs of the entire community, including heterotrophs, from the gross carbon income, and you arrive at net community production. So this is the proportion of production that remains after the ecosystem as a whole has paid its bills, making it really one of our best indicators of carbon accumulation and the potential for export. And then finally, export production is telling us how much of that remaining carbon actually escapes the surface ocean here. So all these processes are inherently linked, but they can become decoupled from one another as a function of oceanographic conditions or individual cell physiology, microbial community composition, or grazing pressures, nutrient remineralization, and even the physical transport of water masses. So the consequence is that just every measurement we discuss contains some degree of uncertainty. We're looking at different biological processes integrating over different timescales. We have different assumptions. So there's a really remarkable amount of diversity in the methodology here. And that's precisely why no measurement is really capturing the entire story. So at the end of the day, which one of these is the best, right? And there's not an entirely satisfactory answer, but it partially depends on the scale of the question that we're asking. Some measurements capture processes occurring inside individual cells over fractions of a second. Others integrate entire communities over hours, days, or even seasons, and others are designed to describe whole ecosystems over hundreds of kilometers. So the methods aren't just measuring different things, they're fundamentally viewing the ocean through different lenses, which is an important consideration because the questions we're asking can kind of determine the scale at which we make our observations. So rather than thinking of these, this is my last slide here, rather than thinking these various methodological approaches as competitive services, I find it more useful to frame them as sort of stations along the same journey. So some methods observe the very first interaction between light and chlorophyll, and others follow the movement of electrons through the photosynthetic machinery. Others measure oxygen through carbon fixation, biomass accumulation, ecosystem metabolism, or as we've covered, the eventual export of carbon into the deep ocean. And none of these are really telling the entire story by itself, but each one is revealing something that others cannot. And this is one of the things that I really appreciate most about this minefield of nuance that we have to navigate in biological oceanography. That gave me trepidation at first, but the ocean is far too complex for any single instrument or any single scientist to completely understand. And progress comes from combining perspectives, different methods, different disciplines, different ways of asking questions, different people, different minds at it. Primary productivity isn't a single event that happens at one moment in time. It's a continuum here, a cascade of interconnected physical and chemical, biological processes operating across vastly different scales, from electrons to entire ecosystems. And every method we've discussed today simply just represents a different place to step aboard that journey. And on large scales, it's all headed in the same direction, but we can kind of interrogate the process at various stops along the way to help us better understand functional differences across ecosystems. So again, it might not be a satisfying answer. There isn't one correct measurement of primary productivity. It's just different windows into the same biological story. And the questions you are asking will ultimately inform where along this journey you should hop on. So all right, with that, I really appreciate you all attending and listening, and I also want to extend my gratitude to Shuva and the TREVOR Platt Science Foundation for this invitation to share my philosophical musings on the art and science of measuring primary productivity at sea. But we have our panel ahead here, but please don't be a stranger. Feel free to reach out via email here if you have any follow-up questions that we don't cover today.